Okay, so now we're going to talk about the digestion of lipids, digestion and absorption of lipids, which is a lot more complicated because lipids are nonpolar. Okay, so they're going to have trouble interacting with our mostly water environments in our bodies. Okay, so let's look at this. So lipids do have the tiniest bit of digestion starting in the mouth using lingual lipase. However, it's very inefficient because you do not have emulsification of that. So I am going to simplify and say in red here that the digestion of lipids begins and ends in the small intestine. Okay, just to simplify that. And you'll see that in a lot of resources. They say that anyway. So we're going to say that lipid digestion begins and ends in the small intestine. Okay, so, and the reason for that is because once you get to the duodenum, that's where you're going to have bile present. What organ makes bile? The liver. Okay, so the liver is going to secrete bile into the duodenum. And that's very important because you need to take these large fat droplets and break them up into smaller fat droplets. Otherwise, you can't get at them using the enzyme lipase. Okay, so it's a two-step process. We need to do physical digestion of the fat droplets using bile. And then we can start breaking apart covalent bonds using the enzyme lipase. Okay? So when we say emulsification, what we mean is we're going to take very large droplets, right? So this fat droplet here, this is made up of a bunch of triglycerides that are all clinging together because they're in an environment, right? So in the intestines or even in our blood that is polar. So all of these non-polar triglycerides are going to cling together in these fat droplets that aggregate together. And so what we need is we need an emulsifier. And an emulsifier, in this case bile salts, it has a hydrophobic side and a hydrophilic side, which is really great because it can act as a mediator between the polar and nonpolar molecules. Okay, so what it does is it surrounds these fat droplets and it effectively takes large clumps and breaks them into small clumps. Okay, so that is called emulsification, breaking large fat droplets into small fat droplets. So at this point, after we have used bile to break things down into their small droplets, have any covalent bonds been broken? No, the triglyceride molecules are still fully intact. That is why we say that emulsification using bile is a physical digestion, not a chemical one. Okay, so to do chemical digestion of triglycerides, we need an enzyme. Okay, so the enzyme that we're going to use is pancreatic lipase. Okay, so the pancreatic lipase comes from the pancreas, and it is going to break apart the covalent bonds here and here, well, here and here, I guess. Okay, leaving one monoglyceride, mono meaning one, so one glyceride head with one fatty acid tail attached. There's your monoglyceride, and then two free fatty acids. Okay, so at this point we have broken covalent bonds. So this is chemical digestion of the triglycerides. Okay, so now we need to talk about how do we get these components through the cells into the blood. Okay, so this just summarizes what we've seen. We have our large fat droplets from our food that we've eaten. We have our bile. The bile emulsifies them into smaller droplets. See how the other dots are smaller. We then use the lipase to chemically digest our triglyceride into two free fatty acid tails and a monoglyceride. Those then reassemble into what's called a mixed micelle. And what we mean by a mixed micelle is a mixture of the bile salts plus all of the nonpolar stuff. Okay, so that'll be the monoglycerides and those free fatty acid tails, plus other nonpolar stuff like cholesterol or fat-soluble vitamins like vitamin D. Okay, so those are all going to clump together in a mixed micelle. Now at this point, we have little clumps that are pretty small and mostly, not always, not entirely, mostly nonpolar. So if you are small and nonpolar, what type of transport do you think we're going to use to get through the cell membrane of the intestinal lining? Small and nonpolar means that they are able to pass right through that phospholipid bilayer into the cells. Okay, so if this is the lumen, right, where the food would be, where we're digesting things, it's going to pass through simple diffusion directly into the cells lining the intestine. Now, once we get inside there, we are going to reassemble, put them back together to make a triglyceride. Now, every semester I get people saying, well, why would we break it apart just to put it back together? Honestly, I don't have an answer to that. I'm not sure why it does that. I think it just does that. Maybe it has to do with the fact that breaking it apart allows for simple diffusion. I don't have a good answer for you. This is just how it is. Okay, so from the pieces, we reassemble back into our whole triglycerides. Now, the triglycerides at this point are having trouble because they are nonpolar. And so we need some help from something that is polar in order to be packaged into and be able to move around in the blood. Okay, so we're going to package it along with some proteins into a lipoprotein that is called a chylomicron. Okay, so we're gonna add some proteins into it. Proteins are polar, package it together, create a lipoprotein called a chylomicron. From there, it'll transfer into a lymphatic joint vessel, so lymph vessel, and that vessel will then join up with the blood. Okay, so let's talk about lipoproteins. Okay, so lipo as in lipid, proteins as in proteins. Okay, so let's talk about why we need to package our triglycerides along with some proteins before they can go into the blood. So when we look at the blood is mostly plasma, and plasma is mostly water, and water is polar. So if the blood is polar, the triglycerides are nonpolar. That means that they are hydrophobic. They cannot interact with the water. And so because they cannot interact with the water, we can't just dump them into the plasma. So instead, what we're going to do is we're going to package them inside of these lipoproteins. And there are several different types of lipoproteins, but we're going to talk about kind of three of them. First, we have the chylomicrons. So the chylomicron, those are the ones that are produced by the cells lining the small intestine. Those are the ones that we just talked about. Once we have a chylomicron, though, the chylomicrons are going to be distributed to the liver. Once they go to the liver is going to repackage them into low-density lipoproteins. Okay, so this right here, VLDL, stands for very low-density lipoproteins. So to summarize this, I'm going to clump these all kind of together as low-density lipoproteins. Okay, these low-density lipoproteins will move around to the body, distributing off their triglycerides, and as they travel through the body, they are going to lose triglycerides and turn into high-density lipoproteins, and the high-density lipoproteins will return back to the liver. Okay, so I have a picture to help with this. It's a bit complicated, so here, let's look at our picture here. So here is the small intestine. The small intestine packages the triglycerides along with cholesterol and all that into chylomicrons. The chylomicrons do go to the muscles and the fat tissue for storage as well, but mostly they're going to go to the liver. The liver repackages them into new, different lipoproteins that are called low-density lipoproteins. the low-density lipoproteins will then pass their triglycerides, give off triglycerides to the other tissues in the body. And as they give off their triglycerides, they will turn into high-density lipoproteins. Okay, so let's talk about this transition here. How do the low-density lipoproteins turn into high-density lipoproteins? Okay, so let's look at that. So looking at this, we're just comparing a chylomicron with a low-density lipoprotein and a high-density lipoprotein. Okay, so looking at this chylomicron, we have a lot of triglycerides. We've also got some cholesterol, and they're packaged inside a phospholipid with some proteins. Okay, this is a lipoprotein. See, lots and lots of triglycerides. They then go to the liver. The liver is going to repackage them. And as they repackage, they become more dense. And the reason they become more dense is because they have a higher percentage of proteins. Okay, so more proteins there. Now, as these low-density lipoproteins travel around the body, they are distributing triglycerides to the other organs in the body. And as they do that, that means that the number of triglycerides and cholesterol is going to decrease. And the number of protein is therefore going to increase. So it's not really so much that they're increasing the amount of protein, but that they're decreasing the amount of triglycerides. And proteins are far more dense than triglycerides. And this is why they say, like when you're trying to lose weight, they say, oh, muscle weighs more than fat, right? So when you're losing weight, you might not feel like you're making much progress on the scale, because maybe you're not losing weight, but you might be gaining muscle. Muscle weighs more than fat because it has more protein than fat tissues. Okay, so it's the same idea here, where as we lose our triglycerides, we are going to increase the density of the lipoprotein, because what's left are the proteins. Okay, so that is why the high density lipoproteins are more dense than the low density lipoproteins. They have more protein, relatively speaking, because they have less triglycerides. They have given away the triglycerides. Once they do that, they are then going to be returned back to the liver. Okay, so let's talk about how this relates to health, because I'm sure you've heard good cholesterol, bad cholesterol. You may have also heard high density lipoproteins and low density lipoproteins. Okay, so the reason this is important for your health is that when the liver receives saturated and trans fatty acids, those are preferentially packaged into low density lipoproteins. Now, the problem with low density lipoproteins is that they are sticky. And because they are sticky, they actually can stick to the walls of your arteries, creating plaques. And these plaques can then sometimes recruit blood clots, and they could lead to things like heart attacks and strokes, and that's not good. Okay, so that's why we say that these are bad for you. Whereas unsaturated fatty acids, those ones are preferentially shunted into the pathway to make ATP, right? So they're going to go into Krebs cycle. So they tend to be converted into ketone bodies instead of being packaged into lipoproteins. So because you are creating fewer low-density lipoproteins, you're going to have a higher ratio of high-density lipoproteins compared to low-density lipoproteins. Because you have fewer low-density lipoproteins, you're less likely to create the plaques and have problems with your cardiovascular system. Okay, so unsaturated fatty acids are better for you because of the way that they're used in your body. The liver treats them differently than saturated and trans fatty acids. Okay, so that's a bit much. Let's see. Which of the following statements is not true? Let's see if you can figure it out. So the answer is D. Triglycerides need to be packaged into lipoproteins because they are hydrophilic. No, they are hydrophobic. They are non-polar. Okay, and then the other ones, A, B, C, those are all true, and they are just testing your understanding that as they travel through, they lose their triglycerides. Okay, so as the low-density lipoproteins contain fewer than the very low density, right? Because what determines the density is how many triglycerides they have, and they give away their triglycerides as they travel through the body. So you go from very low density lipoproteins to low density lipoproteins to high density lipoproteins. Okay, so that's this picture here. Okay, so if you have not done so, please complete the chart, and I have published the answer key right here on Canvas in the Week 6 module. Bye-bye.